Taking just a few seconds to cool freshly laid eggs could add weeks to their shelf life, according to a Purdue University study. The rapid cooling process uses liquid carbon dioxide to stabilize the protein in eggs, thus extending their shelf life.
You can cool eggs many different ways. Traditional cooling of eggs, which would be the eggs you would find at the grocery store, those are typically just cooled under ambient conditions, where the eggs are put into a carton and they're about 105 degrees Fahrenheit, and then they cool over time. It takes about 10 to 14 days for those eggs to cool to refrigeration temperatures. The egg cooling system that we've developed here, based on using carbon dioxide as a liquid that we can spray and create a cold snow, and this snow is about minus 107 degrees Celsius, negative. This snow can be circulated around the eggs, and we're able then to rapidly cool eggs. Instead of taking 10 to 14 days, we can do it in the range of seconds of treatment time.
Eggs cooled under current methods lose the AA grade in about 6 weeks. Keener's study shows that this rapid cooling process can significantly increase this time period.
So typically your eggs at the grocery store, those are traditionally cooled where they're just allowed to cool under ambient conditions. The shelf life on those eggs is usually 4 to 6 weeks. We actually looked at 3 different cooling regimes, minus 45, minus 60, and minus 75 Celsius. And so under those conditions, we were exploring whether or not there would be an impact on our ability to enhance or affect the white of the egg. The egg white contains proteins and water. What we've documented here is we could actually, those eggs, the quality of those eggs would stay longer than 12 weeks. So we've actually identified in this work that if we cooled the eggs with the CO2 process, the quality of those eggs was equal to what they were under the initial. So as if the chicken had just laid the eggs, that quality was maintained for 12 weeks.
An increase in shelf life wasn't the only result of Keener's research.
The other part of the study that we looked at was the vitelline membrane, which is the egg yolk membrane. So you would be familiar with that. When you break an egg out in the skillet, it's what contains the yolk. And so we've documented in this study that the CO2 process that we've been exploring actually will maintain the structure of that membrane longer. And the potential benefit is that it would be a barrier for bacterial penetration. So in essence, the egg safety could be maintained even longer. So to summarize the results of the study, we documented that the vitelline membrane, which is related to food safety, can be enhanced or maintained longer. And then the quality of the eggs, the shelf life based on the quality, can be extended from 6 weeks to 12 weeks.